The final case study that I've chosen for this semester is looking at accelerated venom evolution in a species of viper. I chose this one because it illustrates some of the principles we've been talking about and the application of those to a very specific project. I also just think it's really cool. It's a fairly recent paper. There's also been a lot of other work, and I've provided a link to one other even more recent paper that also does some similar things. It's not required, but if you're interested, you can look at it. I'll mention that here in a little bit. So for this paper, we need to remember the background of the origin of toxins in the squamates. And squamates include snakes and lizards. You should remember that snakes as a group, it's a monophyletic group, but there are some lizards that are more closely related to snakes than they are to the other lizards. So that's why lizards is in quote here. It's not a valid taxonomic group. Snakes are really just highly developed lizards that have specialized in many ways, lost their limbs, and of course toxin specialization is one of them. Not all snakes have toxins. Some of them seem to have lost the early primitive forms of toxicity that then were elaborated a little bit in the Anguimorpha lizards, and then to the most extreme in the vipers. The vipers have some of the most extreme morphological modifications and very diverse and toxic venoms. And then the elapids, which their venom delivery systems are not quite as elaborate. They don't have the hinged jaws, but they're still very toxic and do have hypodermic fangs. But their hypodermic fangs are fixed rather than retractable. So in this study, we're looking at the vipers. Now, so understanding this allows us to choose study organisms. It gives us a foundation and a background, a roadmap for picking organisms that would be interesting. So they chose a study organism from the viper family, the pit vipers, which include rattlesnakes, copperheads, cottonmouths here in the United States, and then pit vipers across the rest of the world also. So this is a pit viper from the Ryukyu Islands. These are islands that stretch between the southernmost main island of Japan all the way down to Taiwan. Okinawa's in the middle, if you're familiar with that. But location's not super critical. But the pit vipers, again, have this really highly developed venom system, massive salivary glands. That's the main reason for the shape of these big muscles surrounding them for injection of venom into their prey. And they can also use it defensively. In addition, and this is not covered in this paper, but it's kind of a cool extrasensory, they do have these pits, thus the name. So that's the nostril right there, that smaller hole. And these pits are basically infrared heat sensors. Their primary prey is mammals, and they're able to sense body heat in a way that allows them to hunt even when there's very low levels of light. And so many of them are primarily nocturnal. Okay, so that's kind of our taxonomic morphological background. Now let's start getting into the molecules. So they were interested in looking at the venom genes, all of them that are present in this snake. And so they're going to take a genomic approach. And so they have a number of questions. So they're interested in whether it is transcriptional variation and post-transcriptional variation in the genes that are there, or whether there was gene duplication followed by diversification of some of those gene duplicates. And I'm going to give you a word. We've kind of, I think, implicitly talked about this, but not explicitly given you this word. If you've had my developmental mechanisms class, some of you, I think, took it as an undergrad. You may know this word, but if not, good word to know, good vocabulary word, neofunctionalization. Okay, so essentially, once a gene duplicates, immediately you've got redundancy. Right? And redundancy can have a couple of benefits, right? You've got a backup copy. You could make a higher level of transcripts because you can transcribe now from 2 copies instead of 1. So there's some benefits to just pure redundancy in certain cases. And so sometimes that redundancy is kept long term. But more frequently, when you have an extra copy, it will either mutate and become part of the non-coding DNA of the gene because you don't need the 2 copies, or more rarely, but very, very importantly for the evolution of gene diversity, it will become neofunctionalized. And so when a gene loses its function and becomes part of the non-coding DNA, if we could still recognize it, we call it a pseudogene. However, if a paralog gains a new function that the ancestral gene did not have, that's neo-functionalization. Big long word, but it's nice. It's fairly intuitive what it means. So in this study, they're going to be looking to see whether with gene duplication followed by diversification of one or more of the copies of those genes led to a new, and in this case, toxic function for that gene. And the reason they're able to do this now is because we have entire genomes. We can relatively cheaply and easily sequence genomes, and not only that, but compare them to transcriptomes, which is an important part of this study, and we'll look at here in just a little bit. So here are some of the questions that they're going to be addressing. First, what type of gene duplication impacted the Venome, the evolution of these proteins that are part of Venom? So they're looking at gene diversity, both in extra copies and then evolution of one or more of those copies. And they're looking at alternative splicing, meaning can we have one gene and have it spliced together in different ways to give different outcomes? So that would be transcriptomic diversity. So genome stays relatively the same, but the product from that genome could be different if we can splice genes together differently. And then they're also interested in how there is a fairly well-supported 2 rounds of whole genome duplication that occurred at the base of the vertebrate lineage. And so we can see, so for instance, important developmental genes, the Hox genes in all of the vertebrates, are found in 4 copies. Those have been, all 4 copies have been kept, and many of them have taken on new or sometimes redundant function. And so they're interested if this 2 rounds of whole genome duplication in the early vertebrates played a role and influenced the evolution of the Venom genes. And then finally, if they're able to locate and then identify where genes are found on chromosomes, they're interested to see if there are any patterns. And of particular interest, so this is a little bit more background, but of the genome of the reptiles. And remember, the group Reptilia now also includes the birds. Birds are just highly modified adapted reptiles. So we're looking at crocodilians, the birds, the squamates. There are other groups also, smaller groups if you've taken herpetology classes you might be familiar with. But so remember, I just want to make it clear, when I say reptilia or reptiles, I'm talking about the new taxonomic designation, not the old one where it was a paraphyletic grouping. So one of the interesting things genomically about the reptiles, birds, squamates, other reptiles, is that they have what are called microchromosomes. So there has been fairly strong selection on a subset of the chromosomes. They have elevated GC content. There are higher levels of recombination, so they're a little bit more mutationally and evolutionarily active because of those trends. And they're also much smaller. And so we've got these 7, I guess 8, if you include the sex chromosome here listed with the Z, 8 what are called macrochromosomes. And then we've got this additional 10 much, much smaller microchromosomes. And so part of this study is to determine whether Venom genes show a pattern. And if so, then that might be an indication of past historical selection pressures that were different on Venom genes than the rest of the genome as a whole. Okay, so this diagram, by the way, came from the paper. This is the one I told you about earlier. Here is the link if you're interested. It goes to some other kind of interesting things we've mentioned briefly in the past, dosage compensation, and they're looking at a rattlesnake and doing some similar things to what this habu paper is doing. But again, I won't ask you questions on this paper. Let's move on and finish up the habu paper. So they're taking this whole genome approach. So this entire genome has been sequenced, begun to be mapped. That's a long, drawn-out process before it's complete, but they've got a rough map of where all the genes or most of the genes are located in the genome. And then in addition, they now added transcriptome data. So I think they took something like 20 different Venom transcriptomes. So they can actually compare the RNA that is present in the salivary glands that are producing the venoms, and then map them back onto the genome to see how that works, to see if maybe there are 4 or 5 different transcripts that all came from one single gene. And so that would indicate some post-transcriptional, maybe alternative splicing or other post-transcriptional processes that yield some genetic diversity. And then as they do that, once they've identified all of the genes from the genome and the different types of transcriptomes, they can begin to get a really nice view of the genetic diversity that is produced in these venom genes. So quick summary. I don't expect you to memorize all this, but just a very quick summary. They identified, I think it was 18 different gene families, some of them which have very, very high numbers in the gene families, and some of them have both venoms and then non-venomous members. So these are the non-venomous paralogs from which the venom proteins evolved. And then they're looking at whether there's gene duplication within these small subgroups and whether there is accelerated evolution. We'll look at how they measured accelerated evolution. Now, they classified these 18 gene families into 3 different larger categories. Ones that had like lots and lots of copies and lots of duplication, one that had fewer copies but still some duplication, and then the ones where we pretty much just have a single venom gene that maybe has some transcriptional variation, right? So we've got these 3 different groups, okay? So that was the result of this comparison of what they found on the genome and the mapping and annotation of the genome versus what they were finding in the transcriptome. And so these 18 genes basically looks like they were part of this early 2 rounds of whole genome duplication. And so we take an original ancestral 18 gene copies pre-venom. So these genes have other metabolic physiological functions, but then we end up with 4 copies. And in most of the cases, 3 of those copies, some of them may have been lost, some of them have also gone through duplication and gotten multiple copies, but these three copies of the 4 that resulted from the 2 rounds of whole genome duplication, 3 of those copies kind of maintained a more ancestral function and never were co-opted and never became part of the Venome. But 1 copy of those did seem, in many of these genes that have taken on venom function, they were co-opted, and that 1 copy then was under very different selection pressures as it gained this new ability, this new function in the squamate reptiles. And these are especially elaborate and very, very diverse and different in the vipers, which this habu snake is part of that group. There's also, this is a little bit of an aside, but there's also a little interesting part of the paper where it says there's also a huge amount of even within population variation and different transcriptional process. So this seems to be a very diverse and quickly evolving part of the snake genome that has been under very, very strong pressure. And that's why we see so many diverse proteins as part of the venom, and it's such an important part of how these pit vipers survive and are able to adapt to their environment. And so within this smaller subclass, we have this really large group that were indicated to have very, very high accelerated evolution, and they've called those category 3 genes. And the interesting thing is that even though there doesn't seem to be as much accelerated evolution, there's still a gene duplication that has occurred in these other ones. So these maybe are fairly flexible genes. When they've duplicated in the non-venomous form, they've taken on new physiological, probably important, but in many cases uncategorized function. So they've taken on new function in the non-venomous groups, these 3 copies that were non-venomous, and then they've taken on more function in the venomous ones. So it kind of shows you, and there are just some genes that are a little bit more flexible, are more widely applicable because of the role that they play in the genome and in the proteome in the living cell. So very strong indication of accelerated evolution in 4 of the gene families, and in 3 of them, moderate amount of duplication. Okay, and so I want to just spend a little bit of time about the tests that they did for looking at accelerated evolution, which is, you know, kind of the hook, if you will, the take-home message and the title for this paper. So the way that they were able to do this is by having a kind of a different baseline. So remember, we talked about how when we're looking for signs of selection, neutral evolution becomes our baseline. But there's a little bit of an issue with that, because if we have a gene or a set of genes that varies from neutral, which is your DN to DS ratio, in this case they're using a Ka to Ks ratio, but that ratio, if it varies significantly from 1, that's a sign of selection. If the value is less than 1, then there's signs for this purifying selection, where it's a maintenance of kind of the status quo. If the value, that K to KS ratio, is much greater than 1, significantly greater than 1, then we have a signal for positive selection, that the gene is evolving and taking on new function. So the difficulty here is, what is significant? How much do we have to vary away from that, being exactly 1? If it varies X amount, is it significant or not? And so that level of significance can be a difficult bar to establish. So what they have done, rather than just saying, okay, our null hypothesis, our starting point is 0, our starting point is 1, right? When dn is equal to ds, what we are going to do is we're going to compare what's going on in the paralogs, the non-venomous paralogs, and sometimes there are multiple non-venomous paralogous copies, versus the venomous paralogs. And again, many times there are multiple ones, so we have a little bit more statistical power when we have multiple ones. And so in each of these 4 different gene families, they were looking at rates of evolution in the non-venomous ones, and then rates of evolution in the venomous ones. Non-venomous ones are in blue, and venomous ones are in red. And so notice, if you were just comparing red and saying, oh look, they're all kind of clustering. Some of them have a little bit of a bias towards the positive side, which is on the upper side of this line that goes from one corner to the other corner of the graph. And so we might be able to detect significance, maybe not at a massive level, but significance here if we're just going off the basic null hypothesis. But notice when we're comparing, okay, here's their paralogs and the pressure, evolutionary pressure that they've been under, compared to the venomous versions, notice that there's a clear, distinct difference, very, very highly significant. And this one maybe is even more so. This one, the PLA2 family and the SP family, are very, very highly significant. But there's significant difference in all of these families. Even here, there's a clear, although not completely disjunct in distribution, there's a clear pattern to where we find the venomous genes versus the non-venomous genes. So really, really strong evidence for accelerated evolution compared to the non-venomous paralogs for many, many of these gene families. So that's a kind of a cool result. And then finally, the last thing I want to highlight, the result from this paper, is the location of these venom genes. So again, remember, we established that there are 10 of these microchromosomes in these viper genomes, and there are 8 of these much, much larger. So about 80% of the genome is made up of macrochromosomes, the larger ones, and only about 20% or less is made up of these microchromosomes. Yet when they began to map these of the venom genes, where they could find a clear location on the genome, more than 50% of the venom and non-venomous paralogs were found in these microchromosomes. So that's a pretty significant, right? When more than 50% are found in only 20% of the genome, that shows a clear selectional bias in the past for location of these genes. And so to some extent, and there's further work that needs to be done on this, we can look at perhaps the macrogenomes of the snake as being kind of more ancestral, carrying on the conserved functions that make snakes very similar to other reptiles or even other tetrapod vertebrates. And then these microchromosomes are almost like a set aside little separate part of the genome that can evolve more quickly and rapidly and take on new distinct function. So maybe we could even look at these microchromosomes as the chromosomes themselves have become neo-functionalized, doing something unique for the squamates and maybe even other reptiles, but certainly here with the venoms, that is making them distinct from other tetrapod vertebrates. And in fact, this result was even more widely supported. I think they said 89% of all of the venomous genes that they were able to locate were located in microchromosomes. And so the quality of those chromosomes, the high GC content perhaps, but maybe even more importantly, the high rates of recombination that can occur in these microchromosomes, favors for this accelerated rate of evolution that there's a clear signal for in these venom genes among the snakes.